mean? So, um, extracting with like ethanol or, some kind of alcohol or actually CO2, I think would be a really interesting one. But yeah, that's kind of what we would do is we would do different extractions on the mushrooms, you know, and basically, take those extractions, do certain experience. I'm trying not to give anything away or
Yeah,
valid-
sure.
validate NDAs, but
Right.
basically, basically break down the mushroom chemically and then see if any of those parts actually had any validity.
Very interesting. So, I mean, I would imagine that your day is consumed, for the most part, working directly with clients on, various projects. How big is Bioinfo Solutions? How many people, work with you? And then also, what does a typical day look like?
Sure. Um, we're pretty small. As you can imagine, it's the computer is mighty, so the individual can do a lot of stuff. I would say, I probably have maybe three or four, that I consistently work with, but doing this for like 20 years, I've gotten to know a lot of people. So I think that's the thing with bioinformatics is that it's not just, like another specialty. It's bioinformatics is huge. You can actually do absolutely everything. You could do, there's transcriptomics, there's proteomics, there's mass spec, there's, looking at the DNA sequence, GWAS, right? There's just so many different areas that there's no way one person can specialize in any of those. So, or all of those. So what I do is I get a lot of people that, are interested in bioinformatics. I do a lot of workshops so that I'll go and what happens is, I'll give a workshop on bioinformatics, and then at the end, I'll have students come to me and say, I really want to do this. I don't want to go in the lab and sex mice, at 5 a.m. for an experiment. I want to just be at home, working and actually doing research there. So I get a lot of students that actually want to help out, and so I work with them. But yeah, a typical day, usually I have a few meetings, talking to clients. I think it's very important as a bioinformatician, I'm going to stress this is to understand the biology,
Mm,
right? I came from the bench, I've worked in labs. I do understand the biology that whenever you do an experiment, all these genes that you're looking at actually have a history, right? They have buddies, they have enemies, within these genes. And to be able to understand that a priori and then put that information together, I think makes a better model than just coming into it and just looking at it as just regular data. Um, a lot of my job, and I say this when I give workshops, most of my job is taking data that taking a lot of data and putting it into a format that I can actually look at. So 80% of my job is probably just massaging the data so I can actually analyze it. So some of it's kind of mindless, you can do this anywhere. A lot of times I'll get bored at home. I'll, go to a coffee shop and, work or, wherever. So bro, it is a lot of, part of it is, when I teach this, have you ever seen the first Karate Kid,
yes.
right?
Yeah.
Movie, right? That kind of wax on, wax off, and he's hating the fence, and he's like, "Why am I doing this?" This is kind of, that's when I get somebody that wants to do bioinformatics, usually I have them, annotate, massage, put together the data, and that's the waxing on, waxing off. But as you put all this information together, what you find is, you start learning more about the study, and that, you know, for me to put together data and to figure out the variables, a lot of times I'll have to go in and actually read the paper. So
Mhm.
you're reading these papers, and if I say I'm working on sepsis, I end up reading like 5 or 6 different sepsis papers, and at the end, I get a better idea of what's going on, and I understand the data sets itself and what it can and can't do, if that makes sense.
Yeah, it makes a lot of sense to me. So the one thing, we probably haven't talked a lot about in different podcast interviews, but so I'm a Lean Six Sigma Master Black Belt as well. And one of the things that I oftentimes would tell people, I'm like, look, the statistics tell you a lot, but the conversations around, like, the data tell me a lot more. You're like, why am I seeing certain things at different
Yes.
times? Why do I see, certain peaks and, workloads and other things, different defects happening? Hey, look, we switched a machine or whatever. I mean, all of a sudden it's like, okay, so now now I understand why we're seeing what we're seeing. But to your same point, and we oftentimes would sort of take that long tail, and chop it off and go, look, I'm not even going to look there, because most of the stuff that I need to find is, in the massive data that I see, in the other end. And so that's what I sort of interpret from a lot of the things that you're saying.
Oh, absolutely. When you see, for me, when you see a paper come out on, say it's a big, transcriptomics or proteomics study, it's like when you see the paper, that's like the tip of the iceberg, right? And then you have all this lovely, gorgeous data underneath, where 90% like. So for me, I look into, these databases, Gene Expression Omnibus, I look at a bunch of, icebergs that haven't been discovered, so absolutely. There is so much data out there, and that, even in a manuscript, even in a long manuscript, there's no way you can describe it all. So,
Mhm.
so yeah, there is absolutely hidden gems in just about every study that you look at. I will also say there's a lot of garbage out there as well. And you don't realize it, it's pretty easy from a transcriptomic perspective to figure out like males and females in a study. And a lot of times you'll look at a study, you'll download it, and the individuals that they say are males and females are not that. There'll be mistakes. And when that happens, you're like, how well can I trust this study? And
Right.
sometimes that happens in, studies that are, been popular, published in really, famous journals too. So
Mhm.
it's crazy.
Yeah, I mean, look, I know that mistakes can happen, but you talked earlier about, sort of collections of data and the fact that there are, places that you can go where there are past studies that were ran and things like that. Do you worry about that? Sort of taking other people's data and studies, and whether or not if they would actually um, be able to be replicated.
Yes, that's a huge issue. I have analyzed people's, the interesting thing, I come in, I have that post perspective, right? Like as you're in as they're in the thick of their experiment, they're just kind of, a lot of times they'll be focused on whatever they obviously wrote their grant for, probably, and so we've got to
Mhm.
definitely focus in on that. But, I'll get the paper, I'll download the data, analyze it, and I'll come up with something completely different. And a lot of
Mm.
times I'll actually reach out to them and say, "Hey, did you know this was in your data?" Sometimes they're very happy that I did that, and they're like, "Oh my gosh, I can't believe that's great," and that might be a potential collaboration. But a lot of times they're very upset that you're actually looking at their data.
Mhm.
You would be amazed at how many researchers out there get very upset with what I do. And I've presented at conferences and had like these older professors stand up and just yell at me like, "You're
Oh, wow.
stealing our data. You're ruining everything."
Aw. It's a shame that that's the case. I mean, I know at least for, I don't know, my PhD research, I was happy whenever all of a sudden people were reaching out, wanting to reuse a study that I had. I was like, hey, this is actually great to know that it lives on, and that, somebody else is finding some use in the thing that I did, that I didn't think that was all that valuable in the end. So
Yeah.
that's a good thing. So
I will say it's more of a, from my end, I get more value out of taking lots and lots of studies. So in a way, they usually typically you don't get mad at me because, they might have their data might be, N of one in, like 10 studies I looked at. So I would say I think that's the best way actually to look at this data is that, kind of a wisdom of crowd kind of thing.